So the first test that we're going to be doing is the amylose hydrolysis using this starch agar plate. Our first step is to sterilize the inoculating loop for 10 seconds and then withdraw and let it cool for about 30 seconds. We're going to mix the source culture well and take the cap off of the source culture and sterilize the lip of it for about 5 seconds. And then we're going to get a sample and then sterilize the lip again before putting the cap back on. And then we're going to open the agar plate clam style and put the bacteria on the plate with a single line streak, and then close the plate. And then we're going to sterilize the inoculating loop again for another 10 seconds and incubate this plate at 37 degrees for 24 to 48 hours. And then after it's done incubating, we're going to add several drops of grams iodine to the top of the bacterial streak and gently swirl the plate to spread the iodine. And so then the next test that we're going to do is the casein hydrolysis using this skim milk agar plate. It's pretty much the same steps as above, so what we're going to do is sterilize the inoculating loop for 10 seconds and let it cool. We're going to mix the source culture well and take the cap off of it, sterilize the lip of it for about 5 seconds, get a sample, sterilize the lip again. Then we're going to open the agar plate clam style and put the bacteria onto it with a single line streak, and then we're going to close the plate and sterilize the inoculating loop for another 10 seconds and incubate this one at 37 degrees for 24 to 48 hours. And so then the next one is the tryptophan hydrolysis test using this tryptone broth. The first step we're going to do is sterilize the inoculating loop for 10 seconds and let it cool. We're going to mix the source culture well and take the cap off of the source culture. Then we're going to sterilize the lip of the tube, take a sample, and then sterilize the lip of the tube again and put the cap on. Then we're going to take the cap off of the inoculating broth, sterilize the lip of that tube for 5 seconds. We're going to put the sample in, kind of swish it around a little bit, and then take the loop out, sterilize the lip of the tube and put the cap back on. And then we're going to sterilize the inoculating loop again and put the cap and take it out to let it cool. And then we're going to incubate this tube at 37 degrees for 12 to 48 hours. And if there is bacterial growth, then we will proceed to add about 10 drops of the Kovacs reagent into the tube and mix gently by rotating the tube in between the palms. So our next test is the urea hydrolysis using this urea slant. And so the first step is to sterilize the inoculating needle for 10 seconds and let it cool for about 30. And we're going to take the cap off of the source slant culture and sterilize the lip of the tube for 5 seconds. Then we're going to take a culture of it, sterilize the lip of the tube again and put the cap on. And then we're going to take the cap off of the urea slant and sterilize the lip of that tube. We're going to take the inoculating needle and push it down about 3/4 of the way into the agar, and then streak along the surface. And we don't sterilize the needle in between. And so after making the streaks, we're going to sterilize the lip of the tube and put the cap on. And then we're going to sterilize the inoculating needle for 10 seconds and then incubate this slant at 37 degrees for 12 to 48 hours. And then the next one is the catalase test. So our first step is to place a drop of the hydrogen peroxide onto the microscope slide. And the lab manual said to use a wooden stick, but I wasn't really sure if that would burn in the micro incinerator. So we're just gonna pretend that this is a loop because I feel like you need to sterilize it. So maybe I'm just completely wrong and I'm overthinking it. But whatever it is, I'm just gonna, because it's supposed to be aseptic technique, I'm just gonna pretend it's a loop and sterilize it for 10 seconds while it is, and then let it cool. And while it's cooling, take the cap off of the source culture slant and sterilize the lip of that tube. And then get a culture of it and apply it to the drop of hydrogen peroxide. And then you want to observe the formation of bubbles, which would indicate the presence of catalase. And then we're going to air dry the slide and then flame, cool, and wash it. And then the next test is the oxidase test. So we're going to allow this oxidase reagent to thaw for about 5 minutes. And then open the sterile swab package and remove it by the handle. We're going to open the slant source culture and or take the cap off of it and sterilize the lip of the tube for 5 seconds. And then we're going to touch the swab to the bacterial growth and sterilize the lip of the tube again, put the cap on, and then we're going to place 1 to 2 drops of the oxidase reagent to the growth on the swab. And we want to make sure not to touch the dropper to the swab or the growth. And then we're going to observe the color change on the swab. And then when we're finished, we put it back into the paper wrapper and into the biohazard bag. So for part 2, the first test that we're going to be doing is the Durham tube sugar fermentation test. And so we're going to sterilize the inoculating loop using the micro incinerator for 30 seconds. And while cooling, we're going to take the cap off of the source culture, sterilize the lip of the tube for 5 seconds, and we're going to use the loop to get a sample of it. And then we're going to take the cap off of the Durham sugar tube and sterilize the lip of the tube for 5 seconds. And then we're going to take our sample and swish it around and then sterilize the lip of the tube again for another 5 seconds and put the cap on. And then we're going to sterilize the inoculating loop for 10 seconds and incubate this at 37 degrees for 12 to 48 hours. And then for the mixed acid fermentation test, or the MR test, it's kind of combined with the VP test. So what we're going to do for those is we're going to use an inoculating loop and sterilize it for 10 seconds. And then let it cool and take the cap off of the source culture, sterilize the lip for 5 seconds, and get a culture. We're going to swish the loop in the MR test tube and then incubate that at 37 degrees for 48 to 72 hours. And after incubating, we're going to make sure that there is growth and then gently mix the contents and take the cap off of this tube and sterilize the lip for 5 seconds. And then we're going to sterilize the lip of this VP test tube. And after that's done, we're going to gently pour about 1/3 of the contents of the MR test tube into this one. And you then close this tube and stuff. And then we're going to add 10 drops of the VPA into the VP, it's hard to do this with one hand, into the VP tube. and also about 10 drops of the KOH into the VP tube. And we're going to gently mix that and put it in the incubator, but we're going to make sure to mix it about every five minutes. And then after putting that into the incubator, we're going to add 20 drops of this methyl red to the MR tube and observe the color change. And after 30 minutes of the VP tube being in the incubator, we're going to remove it and observe the color change. So the next test is the nitrate reductase test. We're going to sterilize the inoculating loop for 10 seconds or so and let it cool for 30. We're going to open the source culture slant and sterilize the lip for 5 seconds, and then take a sample and inoculate the nitrate broth. And then we're going to incubate it at 37 degrees for 12 to 48 hours and inspect for growth. And so if there is growth, then we're going to add 20 drops of nitrate A and 20 drops of nitrate B and observe the color change. If there is no change in color, then you would add a few particles of this zinc dust with a toothpick and mix it well and see if the color changes. And then the next test is the citrate test. So we're going to sterilize the inoculating needle for 10 seconds and let it cool. We're going to take the cap off of the source culture and sterilize the lip for about 5 seconds. We're going to get a culture and stab the citrate slant about 3/4 of the way down there, and then without sterilizing in between, we're going to streak it on top too. And then we're going to sterilize the lip of the tube for 5 seconds, and then put the cap on and then sterilize the needle for 10 seconds. And we're going to incubate this tube for 48 hours at 37 degrees. And if there's no growth, then we would return the slant to the incubator for 72 hours to confirm that it's a negative result. And the next test is the motility test. So we're going to sterilize the inoculating needle and let it cool. We're going to take the cap off of the source culture, sterilize that for 5 seconds, get a sample of it, and then sterilize it for another 5 seconds and put the cap back on. And then we're going to take the cap off of the motility deep, sterilize the lip of the tube for 5 seconds, and then we're going to put the sample that we got from the source culture into the motility deep. And we're going to make sure not to touch the bottom, and we're also going to withdraw it directly. And then we're going to sterilize the needle and then incubate this test tube at 37 degrees for 24 to 48 hours and then observe the results. And the last test is the triple sugar iron test. And so we're going to take the inoculating needle and sterilize it for 10 seconds and then let it cool. And we want to make sure that the agar in the slant isn't dehydrated. And so then we're going to take the cap off of the slant and sterilize the lip for 5 seconds. And then we're going to take the cap off of the source culture, sterilize the lip for 5 seconds, get a culture, and then stab the culture into the butt of the tube, and then streak a single line up the slant. And we're gonna sterilize the lip of this tube for 5 seconds and put the cap on, I forget if I said sterilize the lip of this tube and put the cap on, but if not, then do that. And then we're going to sterilize the inoculating needle also, and then we're going to incubate this TSI slant at 37 degrees for 24 to 48 hours, and then check the tube for growth.